seminar. And I thank Professor Bern Kohler from Ohio State University for accepting my invitation. And I have been following his paper for a long, long time, since when I started thinking about the photochemistry of canonical nucleobase. And he is in Carlos Crespo and another guy, I forget the name. They have authored a very, very important paper in this field. I think everybody is very acquainted with it. So thank you very much for being with us, Professor Kohler, and I'm sure your seminar will be very, very interesting. Then Professor Kohler started his career in Stanford with a degree in chemistry in 1985. And 1990, he got his PhD in physical chemistry in MIT, with studying dynamics and with femtochemistry femtosecond lasers. And in 1993, he moved to ETH for a postdoc period. And in 1995, he got another postdoc research, now in Caltech, California. And from 1995 to 2009, he was assistant professor and got the degree of full professor in the Department of Chemistry at Ohio University. But meanwhile, he moved to Montana State University with his group, and then he was interim department head. In 2016, he returned to Ohio State University, now as professor of chemistry and Ohio eminent scholar. And he is also fellow of the American Association for Advancement of Science, president of the Telluride Science Research Center, and member of the executive committee of the American Chemical Society Physical Division. Among his several research interests, I highlight just a few. He has been working with biopolymer photophysics and photochemistry using laser spectroscopy. He's also and always very, very interested in DNA damage by UV radiation. He's always working with development of photochemical approaches to harnessing energy from sunlight, to study elementary photochemical events in metal oxides and nanoparticles, to understand how light-activate catalysts can produce fuels from water and carbon dioxide, a subject that is very, very popular here in Brazil. So I think most of our students can later talk to you about this subject too. And he's always studying photocatalysts using semiconducting nanoparticles for water splitting and for destruction of organic pollutants in aqueous wastewaters. And he has also received a lot of awards. Some of them I'm going to tell you now. Wiley Faculty Award for Meritorious Research, Cox Award for Creative Scholarship and Teaching, Fellow of the American Association for the Advancement of Science, and the 2017 Inter-American Photochemical Society Award in Photochemistry. And he is the author of or co-author of more than 150 papers, an index age around 43, with a total with more than 7,000 citations. So it's a great pleasure to me and to everybody here to listen to you today. Thank you very much for your for accepting my invitation. And during this seminar, I asked to the audience to mute the microphone. And although I wrote here to make the questions at the end, as you had already listened at the beginning of this presentation, you can ask it during the presentation if you wish. And I'm sure everybody will enjoy very much this period we will be together. So thank you very much. I'm just closing my presentation and you can start yours. Okay.
Listen, I'm very grateful for the kind invitation to present today. I'm very happy that they digital version of me can appear today in Sao Paulo. Of course, it would be much nicer if the real me could be there. So I have never been to Brazil, or as I was saying a few moments ago, I've never been anywhere in the Southern Hemisphere. In fact, I guess I've led a very sheltered life to this point, and I very much hope to visit someday. Okay, so today I'm going to split my time and tell you about our longstanding project investigating excited states of DNA, and then also tell you about newer work where we're interested in the photoproperties of melanin. First, let me start with a photograph. This is our campus. You can maybe see the horizon there at the top of the picture. The ice age was not very kind to Ohio. It left the landscape completely flat. So we have no mountains, but we do have quite nice forests. And of course now here in the Northern Hemisphere, we're starting fall, and so you have to imagine all of these green trees are now beginning to be covered with yellow and red and orange leaves. It's really a very pretty time of year. Okay, so here is a photo of some members of my research group. This is a BC photograph before COVID. So this was actually February 2020. You can see the dates there on the marquee. This is a famous theater in downtown Columbus, Ohio, and we had gathered to watch the ballet perform. This was a presentation of a story that you might be familiar with. It's the story of Alice in Wonderland, a very unusual and quite surreal story. And surreal is also a word I think that many of us use for the last year and a half with this very long and stressful pandemic. But I hope everyone is well and that conditions are improving where you are. Okay, so let me begin by talking about DNA. The motivation for studying excited electronic states of nucleobases and DNA strands is really this biological phenomenon of photo damage. And this is really inevitable of having living organisms exposed to solar radiation. And here at the bottom of the screen, I'm showing the most common photoproduct that's formed in DNA. This is a cyclobutane pyrimidine dimer formed between two adjacent thymines. So you see the two T's, they are able, upon UV excitation, to undergo covalent bond formation that ends up forming this photo lesion, this CPD, and this is genotoxic. So this has to be repaired. Okay, so in bright sunlight, which unfortunately we don't have today here in Ohio, it's a rather cool autumn day, about 10 degrees Celsius with strong cloud cover. But when the sun is shining, then this is sort of a typical rate of photo damage. You form about 10 to 100 of these bipyrimidine lesions per second per DNA molecule, right? And that sounds like quite a high rate. And of course, it's important to remember that DNA, with its 3 billion base pairs, has a very large number of bases, and so in fact this involves a quite small percentage of the total bases in a DNA strand, and yet it's biologically very significant. Okay, so that's the first half of the talk. In the second half, I'm going to talk about melanin, and melanin, as I think all of us know, is the ubiquitous natural pigment. In humans, melanin provides color to hair, skin, and eyes, notably. And in the skin, special cells that are actually part of the immune system. They're derived from neural crest cells. And these cells, known as melanocytes, they're dendritic cells. You can see the tentacles reaching out here and interacting with these keratinocytes, these other epidermal skin cells, and they distribute melanin in special compartments known as melanosomes. Okay, and so lately we've become quite interested in the photophysics and photochemistry of melanin, and I'll talk about that during the second half of my talk. So just to orient you with DNA, in my group we use spectroscopy, so let's look at some spectra. This is UV visible absorption spectra of the 4 canonical nucleobases, A, C, G, and T, and you can see that they all absorb in the ultraviolet below about 300 nanometers with quite strong absorption around 260 nanometers. Okay, so these are the chromophores of DNA. These are the light absorbing parts of DNA, and the entry point for excitation energy that can eventually lead to photo damage. All right, and of course, DNA is not just nucleobases, it's a supramolecular assembly, and so here's a picture of the double helix, and just reminds you about the non-covalent interactions of both base pairing between complementary bases, G with C and A with T, as well as the pi-pi stacking, which I'll often refer to as giving rise to base stacking. Okay, so this photo damage of DNA is strongly conditioned by the light source. Of course, the light source is the sun, but that sunlight has to reach Earth's surface by transiting through our atmosphere. And so the atmosphere actually has quite strong effect on the actual irradiance that's present, and that's what this graph is showing. So first this gray curve, the dashed gray curve, that's showing the mostly smooth black body-like irradiance of our sun at the top of the atmosphere. So that's what you would measure in a satellite. But at Earth's surface, that's the black curve, and so you can see that ultraviolet light below 300 nanometers in what we call the UVC region is essentially completely attenuated. There's essentially no flux of UVC radiation at Earth's surface. And I'm also plotting on this graph, the colored curves are the same absorption spectra for individual nucleobases at upper left, now shown on a logarithmic axis. That's the axis it's also a logarithmic axis I've used to show the solar irradiance, and you can see that the overlap is quite small here in the UVB region. The little bit of solar radiation that is able to leak through our atmosphere is just sufficient to excite the nucleobases really in the tail of their absorption spectrum. Okay, and so we're mostly protected. Okay, that is the shielding that results from our atmosphere, which is mostly due to stratospheric ozone. Eliminating UVC eliminates most excitation, but not all. Okay, and so it's that little bit of UVB excitation that leads to all of this very important photobiology connected with the damage of DNA. Okay, and then an interesting observation is that wasn't always the case. That is, the composition of our atmosphere has changed over historical time, and that's illustrated on this graph. The unit here on this axis is a giga-annum, so this is a billion years, and so you're looking at essentially the entire history of Earth, and for roughly the first half of that history, the atmosphere was is believed to be strongly anoxic with no oxygen and therefore no protective ozone layer, and therefore the rate of DNA damage was particularly high during this ancient Hadean period of our planet's existence. And yet the fossil record is quite clear. The very oldest fossils appear not long after the origin of our planet. So it didn't take life very long to get started. And that means that life initially experienced a photochemical environment that was really extremely harsh. Okay, and that lasted for probably over 1 billion years before this great oxidation event took place. The onset of oxygenic photosynthesis led to the oxygenation of our atmosphere and the formation of this protective ozone layer, and that's of course the conditions that we enjoy today. Okay, so that is a very long time perspective, and I'll come back to that in a few minutes. First, let me tell you about the very first experiments that we did just over 20 years ago here at Ohio State University was to use femtosecond pump probe spectroscopy, specifically femtosecond transient absorption technique, to measure the excited state lifetimes of monomeric DNA and RNA bases. Okay, so we measured those accurately for the first time using as I said, this femtosecond transient absorption technique. So we use a femtosecond UV pump pulse to transfer population from the electronic ground state up to the first excited singlet state. That's a singlet pi star state, and then we can come in a variable time later with a tunable pump pulse. In this case, I'm showing a pump pulse centered in the visible at 570 nanometers, and we can now catch that excited state population and promote it to a higher lying excited state. So this produces a positive signal, this excited state absorption, and you can see that in the data on the right. And now as the population relaxes back to the ground state, the excited state absorption signal goes away, and this allows us to determine very precisely the excited state lifetime. So we did that for all of the bases. I'm showing data here for adenine, and you can see that the lifetime is subpicosecond, just 200 femtoseconds. This is really extraordinarily short lifetime. You know, recall that many organic compounds which are fluorescent have excited state lifetimes typically of a few nanoseconds. And so this is orders of magnitude faster. This ultrafast internal conversion is what we can follow very precisely using this ultrafast laser spectroscopy. And the point is that the population is returning to the electronic ground state here in just hundreds of femtoseconds by a mechanism that I'll describe shortly. Okay, well, there's a second kind of experiment that's very informative. We can replace that visible probe pulse with a probe in the ultraviolet and now do a UV-UV experiment. And this allows us to probe ground state bleaching. Okay, the pump pulse depletes the population in the electronic ground state, and now with less population in S0, there's less population to absorb, and so we get now negative signals. This is a difference technique, and so this depletion results in negative signals that allow us to see how quickly the electronic ground state is repopulated in its lowest vibrational levels. Okay, and so it's very interesting to observe that these bleaching signals relax considerably more slowly than the decay of the excited state absorption. You can see that goes to 0 here in just 1 or 2 picoseconds, but the bleach signals here recover most quickly in water, but you can see it still takes, you know, a little more than 10 picoseconds for the bleach signal to really recover, and those dynamics are even slower in organic solvents, methanol and acetonitrile. And so what's happening here is that the process of internal conversion or non-radiative decay, this is, we should really think about this more as a kind of a horizontal process that takes place isoenergetically, and without a photon to carry away the absorbed photon energy, after UV excitation, we have on the order of 5 electron volts of energy to distribute among the vibrational degrees of freedom of the nucleobase, and that energy corresponds to a vibrational temperature of as much as several thousand Kelvin. Okay, and so that's very hot vibrationally. It's obviously out of equilibrium with the surrounding solvent molecules, and so this then drives this process known as vibrational cooling, where this very hot chromophore following internal conversion has to reestablish equilibrium with the solvent. Okay, and so that's what these negative going ground state bleach recovery signals are sensitive to. They monitor vibrational cooling, and you can see that that is taking place most slowly in acetonitrile, and then it becomes progressively faster as we increase the hydrogen bonding character of the solvent. And finally, in water, with its dense network of hydrogen bonds, this is really optimal for very quickly distributing energy intermolecularly, so intermolecular vibrational energy transfer to the solvent, which is responsible for this vibrational cooling. Okay, so the kinetics I showed you there, I want to show you data now with a broadband point of view. So this is transient absorption data as a function of the probe wavelength now. That's the horizontal axis, and delay time is shown on the vertical axis. And this shows quite nicely how the excited state absorption, which we detect primarily at visible probe wavelengths, is over very quickly. Again, that's the 200 femtosecond decay time to reach the ground state. But then in the near UV, we see this hot ground state absorption. Okay, so all of that energy produces a strongly redshifted absorption spectrum, and the vibrational cooling that returns the nucleobase to thermal equilibrium with its surroundings takes longer. Okay, and that's what's responsible for the slower kinetics seen in the ultraviolet. All right, and so this is the data here for the mononucleotide of adenine. So how does this work? How do the nucleobases undergo ultrafast internal conversion? Well, I know this is a computational photochemistry seminar, and so this is my opportunity to congratulate all of those people that have been working on electronic structure calculations. This is really how we have come to understand the how of non-radiative decay by DNA and its nucleobases. The experiments really just time things. They measure how much time is required to make these state transitions, and we're really blind to molecular coordinates. So that's where we looked at calculations to understand the mechanism behind non-radiative decay. So here I'm highlighting calculations from Spiridula Matsika's group made many years ago. Although it's an old paper, it's still a very fine illustration of just how non-radiative decay takes place. So this horizontal coordinate here is a multidimensional molecular coordinate that reflects the loss of planarity in the molecule. The point is that upon electronic excitation, these planar aromatic molecules tend to experience a loss of aromaticity. We're exciting a pi to pi star excitation, and so we put an electron in a pi anti-bonding molecular orbital, and that makes it easy then for these to undergo ring deformation. Okay, and so these ring deformations are very typical. And you can see that causes the excited state energy to decay, but the aromatic ground state is much more unhappy about this loss of planarity, and so its energy rises very steeply until these two surfaces meet eventually in a conical intersection. Okay, and it's these conical intersections that are really the ubiquitous players in deactivating excited states of the monomeric nucleobases in just hundreds of femtoseconds. Okay, so this calculation shown here for uracil is nonetheless quite typical of what's seen in the other nucleobases with these ring deformation conical intersections and ring puckering CIs. These are often called, which lead via barrierless pathways to deactivating CIs. Okay, so you can think about it as it's almost as easy as falling out of bed. There's just no barrier put in the way, and from the Frank Condon excited state point where we initially appear on the S1 excited state surface, it's all downhill to the CI, giving rise to femtosecond lifetime. All right, and then just an observation: the ultra short lifetimes of the canonical bases, that's the blue squares, seem to be a little bit of a special functionality when we look at modified nucleobases, nucleobase derivatives. Then in many cases the lifetimes become longer, and this was very interesting. This notion that we have almost optimal rates of non-radiative decay in the canonical bases. The only derivative here that has even a shorter lifetime is hypoxanthine, which turns out to be a minor RNA base. So we can maybe include that in the set of organic structures that nature selected for DNA. So this of course is very important because ultra short excited state lifetimes are correlated with high rates of resistance to photo damage, right? If you want to avoid photochemistry, the very best thing to do is to eliminate the electronic energy on a fast time scale. So that then leads to the following evolutionary perspective, the notion that when we look at photophysical properties of DNA, we may actually be looking back in time, if you will, at properties that were likely quite important in the selection of the particular organic molecules that make up DNA. Okay, so when I think about a primordial soup, then on any planet like ours that's near enough to its star to have liquid water, there's going to be a very rich photochemistry. I mentioned already the first roughly 1 billion years of Earth's existence with much less photochemical filtering in our atmosphere, and consequently there would have been a very, very aggressive and very rich ultraviolet photochemistry. So we can think about that primordial soup as experiencing constant processing by UV radiation. And so when you think about the kinds of molecules and chromophores that could come into existence, then that creation of molecular structures is going to be counterbalanced by their photochemical degradation. So you can cycle around between photoassisted synthesis and photodestruction, and of course this suggests that there's a strong photochemical selection pressure for intrinsically photostable assemblies and building blocks. All right, and so that's kind of our idea, is that the an important dimension for this primordial soup paradigm is that this soup, certainly in light exposed environments, is going to be rich in intrinsically photostable building blocks, and so that's of course really the precise photophysical functionality we see in DNA, the ultra-fast internal conversion. That's the same functionality found in absorbing sunscreens, and so DNA seems to be quite well protected to resist photo damage, and this is what we call intrinsic photostability. All right. Well, when the DNA bases are assembled into strands, then things can change quite dramatically. So I'm showing here transient absorption results for an 18-mer, so a single strand made just of adenines, and I'm showing transient absorption in the same spectral window, but I had to use an extension here of my time axis, and in fact it's logarithmic in the upper block because the K rates are drastically slower in this strand. So this was very surprising, and so we published this surprising result in 2005, and with all of these conical intersections that are there for the monomeric nucleobases, it was sort of thought at first that in these strands it would be that much easier with all of their molecular degrees of freedom to access those deactivating CIs. Okay, and in fact, to our surprise, excited state lifetimes are observed to be orders of magnitude longer in these strands. Okay, these longer lifetimes are gated very strongly by base stacking, and that was demonstrated in a couple of ways. We can first destack a dinucleotide by dropping the pH and putting a proton on each of two residues, and that Coulombic repulsion will cause a stacked dinucleotide to unfold. Alternatively, we can put that dinucleotide in a methanol or alcohol-rich solvent. Alcohols are denaturants that disrupt pi stacking. So that also completely eliminates the slow or long-lived component that we see in stacked nucleobases. Quick observation about stacking is that this picture, which I just presented, really isn't a very accurate one. This is not a probable conformation that you're going to find for an adenine 18-mer in aqueous solution, and that's because this pi-pi stacking interaction is really very weakly stabilized. Okay, it's only slightly more favorable than thermal energy, and of course that's a very important part of any biomolecular system. Especially for DNA, it's important that it can be readily folded and unfolded and read out by proteins, and so that requires structures that are quite weakly stabilized. Okay, so this gives rise then to some very interesting interplay between structure and dynamics, which we've explored over the years. I just want to tell you quickly about the nature of these long-lived excited states in DNA strands. These are assigned to charge transfer excited states that have the character of radical ion pairs produced by electron transfer from one nucleobase to its pi stack neighbor. Okay, so that's the cartoon here, that UV excitation causes an electron to be transferred from this O base. O here is an oxidatively modified guanine, known as 8-oxoguanine. This is a common damaged photoproduct, and we chose it for this study simply because it's quite a good electron donor, and so in this dinucleotide, excitation causes an electron to be transferred from the modified guanine to the adenine. That happens actually faster than our time resolution, happens in less than 200 femtoseconds, forming this ion pair, which then subsequently decays with a lifetime of about 60 picoseconds. Okay, so the long-lived lifetime that shows up not just in DNA dinucleotides, but also in longer strands, is assigned to charge recombination by these CT excited states. Okay, and this was worked out with a technique known as time-resolved IR spectroscopy, TRIR, where we combine electronic excitation with a UV pulse with vibrational probing in the mid-infrared. So it's a time-resolved vibrational spectroscopy that is very structure specific, and we're able then to readily differentiate and oxidize nucleobase from a neutral or from a reduced base. And so it's the characteristic shifts in vibrational modes of the nucleobases that happen upon electron transfer that allowed us to make these assignments. Okay, so this can explain the very strong gating seen by pi stacking. Recall that we don't see these long-lived lifetimes unless the bases are closely stacked, right? So that corresponds to a distance of about 3.5 angstroms in DNA, and at that short distance, we can think about typical rates of electron transfer in either proteins or DNA, which take place at fixed distance. So these distance-dependent rates show an exponential distance dependence, reflecting the exponential fall off of the wave function of electron donor and electron acceptor, and so you can see that to be competitive with deactivating CIs, we have no more than a few hundred femtoseconds to have a competitive decay channel, and so simplified argument, this electron transfer rate is only going to be competitive when we encounter these very short distances that we find at contact in these pi stack structures. Okay, and then very quickly about the double helix, we've looked then at what happens when we associate two strands by hydrogen bonding. And so this cartoon shows that we still have these intramolecular CT states or intrastrand CT states, but now we can form radical ion base pairs. And I'll show this cartoon again. We form a radical base pair with a positive charge and one with a negative charge. And in the anionic base pair, we observe a proton move from one strand to the other. That's energetically favored in the radical anion base pair. It's only very weakly favored and not observed in the cationic base pair, and so that's the only proton that we see move. But that's something that we've explored then in a variety of model systems. This is the world's shortest DNA duplex containing just two base pairs, and this was of interest to us because this is also almost the limit, or at least was the limit about 5 years ago, of the largest system that you can treat at a high level of theory fully quantum mechanically. So this tetramer of bases or pair of base pairs is, of course, so the minimal system for seeing the photoinduced proton-coupled electron transfer decay process that we described in DNA, and so it's also the minimal size system that you want to study computationally. So these were experiments then on a system that's fully commensurate with the precise system studied quantum mechanically, and it's interesting to point out that the dynamics here are very similar in this minimal two base pair system as they are in this alternating a teamer that consists of a full 18 GC base pairs. Okay, and those similarities are really indicating that these excitations are very localized. They don't delocalize or spread through the strands, and that's probably an important part of protecting DNA, of eliminating the threat of damage. So what we see is that excitons, initial excited states in DNA, self-trap very rapidly. They form states which you could call excimers, states with high CT character or CT excited states. Many of these descriptions are used synonymously by workers in the field. This happens in less than a picosecond, and then now we have quite longer lived excited states that might be worrisome from the standpoint of photochemical damage, and yet we think that these ion pairs, based on observations made to date that they simply decay with very high quantum efficiency back to the ground state. Okay, so this may actually be photoprotective. The fact that these DNA base pairs are able to separate charge through these stacked through base stacks. You can think of this as a continuous sequence of donor acceptor junctions almost, and that gives rise to these strongly bound ion pairs, and it appears that electron and hole simply can't escape, and their recombination, of course, eliminates the threat of photochemical damage. Okay, so let me move then to the second theme here, which is to tell you a little bit about melanin. I mentioned how most people know that it provides the beautiful shades of human skin, but it's really ubiquitous. It's found in essentially every organism in the tree of life, plants included, fungi. It's responsible for bird feathers, and of course the dark ink of cuttlefish is also essentially pure melanin polymer, and finally enzymatic fruit browning, non-enzymatic fruit browning that you see in avocados or ripe bananas. This also produces melanin-like polymers. For spectroscopists, it's really intriguing. This is the absorption spectrum. It really doesn't look like absorption spectra of organic molecules or materials. It simply decays monotonically from the ultraviolet all through the visible, and in fact extends quite a ways into the near infrared as well. So melanin looks black as a result of this. That obviously makes it interesting for applications like solar energy conversion. We're able to take advantage of a very large portion of the solar spectrum. And so that's made melanin of late quite interesting for applications such as energy conversion and storage. And of course, it's a natural material, so the interface or intersection between electronics and biotechnology, field known as bioelectronics, has been very interested in this polymer. Well, here's the problem with melanin. We don't have a structure. There's no chemical structure, no microscopic structural information about melanin, despite all of the remarkable tools that chemistry has. We just don't know what this is made of. So that makes structure function studies very, very difficult. Without a structure, there can be lots of arguing about function. So we're all a little bit blind to what melanin is really made of at the atomic level, and that kind of gives rise to a situation like the blind monks and the elephant, where everybody sees something a little bit different. And melanin certainly is highly multifunctional, and this has led to a lot of very interesting debates about how these functions are actually established. Right, so it's a natural material, so there is of course a biosynthetic pathway, and here I'm summarizing kind of all of the details that have been worked out. This melanin polymer is formed by the essentially the combustion in air of tyrosines, and the enzyme that is important here is tyrosinase. So tyrosine plus tyrosinase give rise to melanin. And you can see a few intermediates that have been identified, and this picture here is ending with sort of the last recognizable intermediates, and they are the two compounds shown here, 5,6-dihydroxyindole, or DHI, and its carboxylic acid form, DHICA. And so these two dihydroxyindoles are kind of the last reference point we have, and therefore melanin is been discussed as a polymer, a covalent network of joined DHI and DHICA units. But there's been a lot of debate about what the precise assembly motifs are. Are they predominantly linear chains or cyclic structures? And is melanin even a polymer? That is it high molecular weight polymer with perhaps extensive cross-linking? Or a more recent point of view is that melanin might simply be a supramolecular material consisting of much smaller planar oligomers that are simply extensively aggregated in a graphite-like organization. Right. So that's kind of the quick background on how much we don't know about melanin. There really is a very large black box here, which is obscuring the ultimate structure of melanin, and so we can it turns out we can prepare very good melanin mimics using small molecules like L-DOPA. This important neurotransmitter is a great melanin former, and we can make so-called synthetic melanins. And so here's a photo of what this looks like in our lab. So you can begin with a transparent solution of L-DOPA. This absorbs only in the ultraviolet, and an alkaline aqueous solution. All you do is wait, and 6 hours later you see this stark black solution of this synthetic melanin that's been formed. Okay, and so that's the remarkable transformation that is taking place. This autooxidative polymerization is transforming precursors into melanins. But sadly, these synthetic melanins have the same problem that the natural ones do. We also don't know their structural information, and a big part of why is because this whole synthesis happens quite differently than the synthesis of polypeptides and polynucleotides. Okay, for proteins and DNA, enzymes really exert very extensive control on the sequential and iterative assembly of small building blocks into the macromolecules, and that's not the case with melanin. Instead, tyrosinase, the enzyme that's involved, simply makes a quinone, an orthoquinone, a very reactive form of quinone, and that acts a little bit like a match in a fireplace. Okay, that simply initiates reactions, which in fact are very reminiscent of combustion reactions, ones that involve radicals. They occur at diffusion limited rates, occur very rapidly, and consequently there's a lot of kind of radicals gone wild with both covalent and rapid covalent and non-covalent self-assembly that gives rise to the final polymer. Okay, and that produces structures that increasingly are seen as probably being very similar to ones in man-made carbon nanomaterials, like carbon dots or graphene quantum dots. So that's an interesting overlap. And finally, another perspective, melanin is really a member of the very large family of polyphenolic compounds. These include the humic substances in soil, dissolved organic matter in water, even the aerosols, carbon aerosols produced by burning of plant material. All of these make nanoparticles with very similar optical characteristics as melanin. So there's some very universal properties about the chemistry that we'd like to get at. Okay, so in just a couple of minutes that I have remaining, I just want to illustrate quickly some of the things that we've been trying to do as spectroscopists, and I'll focus exclusively on our top-down studies, where we begin with a synthetic melanin, and we try to answer questions spectroscopists would like to answer, like what is it that's making melanin black? You know, what are the chromophores, and can we use spectroscopy to learn about how these chromophores possibly interact with one another? Can we learn about the electronic coupling? Can we learn about factors that might influence or tune both the transition energy and dynamics of these interacting chromophores. Quickly, a little bit of language. Many physical chemists are familiar with multichromophoric systems or interested in multichromophoric systems. The light harvesting complex is a great example of this. But here, multichromophoric means the same chromophore, multiple copies. Okay, many repetitions of bacterial chlorophyll that gives rise to these lovely ring structures with their interesting excitonic properties in light harvesting complex. For the melanins, we think that multichromophore really means multiple chromophores of different types, okay, that they're chemically distinct absorbers. That's a model proposed by Paul Meredith in the field that's come to be known as the chemical disorder model, okay, that concept that this broad absorption spectrum is really made up of overlapping spectra by many components. Okay, so to explore that, we did again femtosecond broadband transient absorption spectroscopy. I'm showing that here. You can see there's positive signals, excited state absorption that decays very rapidly in just a few picoseconds. Most of the signal is lost, and we're looking here at a very broad window from the ultraviolet all the way through the visible to the near infrared. So this is the broadest view to date of excited state dynamics in a synthetic melanin polymer. From the rapid decay, we can learn about the very efficient conversion of light energy to heat, these photothermal properties that become quite interesting of late in many organic systems. And so melanin is a natural sunscreen, right? That's obviously it's one of its biological functions in skin, and we can watch this energy be transferred in a few picoseconds to solvent modes. But what we were able to do, melanin absorbs everywhere, so we can excite it everywhere. And so we use the tunable pump pulse to look at how the sample response changes with different excitation wavelengths. So my excitation wavelength here, I'm showing for 4 different wavelengths: 300, 400, 500, 600 nanometers. Along the top axis, that's the spectrum of the pump pulse that's exciting the sample. The gray curve in every panel, that's the transient absorption spectrum seen when we excite at 300 nanometers. And that's just for reference. Then in color, I'm showing the excited state absorption that we measure using the pump wavelength shown in each of these panels. And you can see that there's a difference in each case. Here with visible excitation, you can see that a large dip appears roughly centered about the excitation wavelength of the femtosecond pump pulse. And that dip or hole, this is in fact what we call a spectral hole. This is transient spectral hole burning here, where we're seeing something that's really quite unusual in molecular photophysics. Generally, for small molecules especially, it really doesn't matter where you excite. You always see a common response. If there's bleaching, then you see bleaching everywhere that the sample can absorb. Melanin, however, we're seeing something very different. We're only seeing bleaching in a narrow window about the pump wavelength, and that's an indication that we have a heterogeneous sample with a broad distribution of chromophores. Okay, so we can isolate those spectral holes and look at their widths, but the bottom line is that this is strong evidence in favor of this model of heterogeneity, this chemical disorder model, suggesting that there are many different chromophores that we can selectively excite. Well, okay, I'm really not going to have time to go into this, but I just want to point out that what's even of greater interest than this transient spectral hole burning phenomenon is the dynamical information that we can extract through ultrafast spectroscopy. So we can learn about the dynamics, and that's turns out to be much richer in its information content than just seeing these spectral holes. And just to give you a sense of that, energy transfer or energy migration is famously important in light harvesting complex. This is a kind of a cascade where energy moves from higher energy absorbers to lower energy traps, and so this is again very important in light harvesting, and it would be expected then to give rise to some dynamics in the positions of these holes, and yet our signals don't show that. Previous slide, you just see that these decay uniformly without motion, and that can be explained by either chromophores that are fully uncoupled from one another, but we actually don't think that applies, and instead we favor this model in which these excitations are giving rise to self-trapped excitons, perhaps a little bit like the ones we see in DNA, charge transfer excitons formed on ultrafast timescales. Okay, and so this is in fact inspired a bit by things we see in DNA, and so we're beginning to explore paradigms where we think about putative melanin chromophores that are pi stacked with one another. There's plenty of evidence from electron microscopy and some other experiments that these graphitic-like pi stack structures are omnipresent in melanin materials, and of course that pi stack geometry, as we see in DNA, is an environment that's very conducive to ultrafast charge transfer, which then of course could be coupled to proton transfer as well. So that's something that we are continuing to explore now, also with multidimensional spectroscopy. That's really something that's very strongly suggested by this chemical disorder model. The fact that you can differentiate absorbers by their excitation wavelength really raises an exciting possibility of now incorporating an additional dimension where one would selectively excite these sub-ensembles and then probe in some additional dimension. And so we published last year a paper in Nature Communications describing just such an approach, a kind of a poor man's version of two-dimensional electron vibrational spectroscopy, a technique we call ultrafast vibrational fingerprinting. This was science carried out by a very talented postdoc, Chris Grieco, who's now a faculty member at the University of Auburn. But this is, I don't really have time to go into very much of the details here, but this is beginning to reveal correlations between vibrational electronic degrees of freedom in melanin polymers. So just to kind of summarize work that has only been going on for about 2 years now in my lab, we are applying ultrafast spectroscopy to try to learn about these mysterious chromophores in melanin, and we've been able so far to show this transient spectral hole burning phenomenon that provides very direct evidence of the chromophore heterogeneity that's present. And now we're just beginning to use some multidimensional spectroscopic tools to look at how we can learn more about electronic coupling and dynamics among chromophores. So I'll be very excited to tell you more about that. I think there's a lot that we're going to be able to learn. Finally, let me acknowledge the many excellent students, postdocs that have contributed to this work, and bolder a few of the names of people who contributed most strongly to the work I've talked about today. Most of the funding for this has come from the National Science Foundation, and I thank our collaborators helped with this project. And finally, I'd like to thank you very much for your attention. As far as I know, everyone's been very quiet. Hopefully, a few questions then that you've saved for the end, and I'd be happy to answer those. Thank you very much.
Thank you very much. Very nice talk. Very interesting subject. Thank you. So please, questions now. Well, I think I'm going to open a section. Oh, okay, Mauricio, please go ahead, Mauricio.
Hi, that was an incredible presentation. Can you hear me?
Yes.
Yes.
Okay. I was wondering about the charge transfer states in DNA. There's a lot of theoretical work that I know mostly from the early 2000s about charge transfer in DNA. Do you are they sort of a locked states, or do you see charge transfer between the bases, or what's? I guess the question is, what's the range of charge transfer? Is it between base pairs, neighboring base pairs, or do they move around? Can you get that information?
Right. So that's a really lovely question, and you're referring to very rich body of work looking at charge transfer and charge transport in DNA strands.
Exactly.
But it's important to point out that in that work, experimentally and computationally, one is looking at just an excess electron or excess hole, and so never a pair, never an electron hole pair, and so that's a very important difference, because for example, the hole transport between guanines, where I imagine a hole moving from G to G. This is a thermoneutral process. So delta G is 0 in this case, and so that's very different than a CT exciton or CT excited state or ion pair, where I now have an electron and as well as hole. So I just point out that the thermodynamic driving force is extremely different for that electron hole recombination. It amounts to several eV, in fact, compared to the driving force for electron transfer from one base to another. Okay, so that difference, I think, plays a big role in making dissociation of this ion pair extremely difficult. That's what I hinted at the very end of the DNA talk, is how we think, although these have much longer lifetimes than monomer nucleobases, the photochemical consequences seem to be minimal, and that seems to be because it's too hard to pull the charges apart. They're just created at 3.5 angstroms, and then they experience very strong Coulombic bonding. So we have not yet seen any evidence of actual transport, where an electron hole would escape and now be able to move through the strand. But it's not to say that it's not happening. It's just that it's probably unlikely, and therefore a rare event that would then also be hard to observe experimentally, because it doesn't happen very much of the time.
Okay, thank you. Very nice.
Okay. Thank you, Mauricio. More questions? Okay. So I think in 2 or 3 previous slides, you showed us some molecules. I think if you please return 2 or 3 slides.
Okay, and so you want me to go back a couple of slides here?
Yes. These
ones, or
one more? Well, I think one more. Yeah, here. Yeah, okay. Do you think that these molecules are the simplest models for melanin, or do you have structures that we can use as models for melanin?
Yes, so these are the favored models. As I mentioned, we can follow the biosynthesis up until the point where one sees these dihydroxylated indoles. And so there is good evidence that these are the basic units, but there's a lot of possibilities for how they could be joined. They can form covalent bonds at a number of different positions, and they can even then create some cyclic structures like you see over in the cartoon on the right, this two-dimensional sheet-like structure. And of course, this is also suggested a little bit by electron microscopy, which shows evidence of graphitic-like structures. So this is sort of another point of view. The reality, though, is that we don't know precisely, and so I talked today just about the top-down approach, but I here in my lab we're also working bottom up, as others have done as well, and what we're doing there is taking these dihydroxy indoles and assembling them, in a controlled fashion into dimers and small oligomers in order to look at their properties. And so it's really a very rich and complex system. You have this unit, which by the way is like indole in some respects. It is a good electron donor. It can be ionized readily, and then it also has this catechol-like functionality that makes it very redox active. And one has then these quinoid or quinoidal structures, and one can cycle between quinone, semiquinone, hydroquinone forms. And in fact, that's an extremely interesting property of all melanin polymers, is that they're paramagnetic. They support a steady state population of radicals. And so there's some very rich radical chemistry that's associated with melanin. And of course, that is very important part of the antioxidant properties and radical scavenging properties that are thought to be significant. So, I can't give really much of an answer to just say that these are artists' renditions, and really nobody has precise information about what the chromophores of melanin are. And again, that's a reason for a lot of interest in bottom-up studies.
Okay, thank you. More questions, please? No? Well, so I have another one, but it's the question I have now is not part of this subject. I don't know if I can ask you now. Or well, it's about. I saw in your webpage that you are working with metal-mediated base pairs, especially with silver. Okay? Yes. So, what can you tell me about the role of silver in the spectroscopy of these compounds?
Yes. Well, yes. This is very interesting. Metals, of course, are very important for DNA structure. DNA is a polyanion, and therefore you always have these positive charges, and we often forget about them. But different metals can interact in different ways, and some are quite important for forming some very interesting supramolecular structures, such as G quadruplexes, guanine quadruplexes, or I motif DNA. And so the nucleobases turn out to be wonderful ligands. They have all of these heteroatoms which can interact strongly with metals, And so it turns out there's very rich self-assembly possibilities. So there's really a whole supramolecular chemistry, and we discovered this a little bit by accident. We thought that we could sort of stick single metal ions onto single bases and then look at how that metal ion binding would affect the ability of the excited state to decay through a conical intersection.
Okay.
Right. We wanted to see the effects of metal ions on dynamics involving conical intersections. However, those experiments turned out to be impossible, and the reason is that for really most cases we've looked at, these metal ions, and especially silver, likes to template a much larger structure. You form coordination polymers, and these coordination polymers in most cases have unknown structures. They form sort of amorphous gels or hydrogels. A few cases there are some crystal structures that have been worked out. And what's interesting is that you see some of the same motifs that you see in natural DNA strands. So for example, if we take adenine and silver nitrate, we make structures, we make nanofibers that we can see in atomic force microscopy experiments. And those nanofibers have a great deal of pi-pi stacking among the adenine units. And we know that because we see very similar decay dynamics to what we see in polyadenilic acid in a regular DNA strand. Okay, so what is challenging there is really the same challenge with melanin, and that is how far can you get as a molecular photophysicist or computational chemist if you don't know the structure.
Yes.
so that's this is a challenge about supramolecular photophysics. Nature has ways of making very, very complex structures, and of course our atomistically to understand this atomistically is very diminished if we don't know those structures.
Okay, thank you.
Sorry, my lights turn off automatically there, so I wave my arms.
Okay.
I'm still here.
Anybody else wants to ask?
But Antonio, I'd be happy to talk later with you more about the metal ion binding. I have a lot of results that we could discuss. Some published, but most unpublished. And if that's an area that you're interested in computationally, I'd be very interested in talking about that.
Okay, thank you. I will send you an email and we can arrange another.
Great.
Okay, thank you very much. So, if there is no other question, I just want to thank you again for this nice talk and for being with us this morning. And thank you, everybody, for being with us. Our students are very shy. They don't like to ask something, to ask much. I don't know why. But I'm sure they learned a lot from you today. Thank you very much.
Okay, great. Thank you very much. It was wonderful.
Okay, have a nice weekend.
Sometime in Brazil today, virtually. Okay.
Thank you very much. Bye-bye, everybody. I will send you an email. So.
Okay, thank you. Thank you again.